Hi, this is Paul Epstein. I'm going to talk here a little bit about the mechanism of esketamine action for the last part or last video of our recent ILO. Um, okay, what I show first, I'll show some of the slides, figures rather, from the Ron Dumond's science paper from 2012. He's part of a group from Yale University that did a lot of the early work on ketamine, showing that it's an antidepressant and trying to figure out how it worked. And What we see on the first slide is that what he shows is that ketamine can rapidly induce synaptogenesis and reverse the synaptic deficits caused by chronic stress. He uses a mouse or animal models of depression, which can be induced by chronic stress on the animal. So the fact that ketamine acts rapidly within hours, it makes it completely different from all the other antidepressants we have, which take weeks. And so based on the actions of how ketamine works, he came up with his own version of a hypothesis for depression, which is shown on the next slide. And he says that depression, in his view, is caused by the disruption of homeostatic mechanisms that control synaptic plasticity, resulting in destabilization and loss of synaptic connections in mood and emotional circuitry. And looking at this further, what he shows in his paper is some examples of what happens when you stress a mouse for a period of time. And so this next slide is an example of a mouse that's been stressed for, I think it was 30 minutes a day for 7 days. And in the figure on the very top, he was looking at the dendrites in density and the connections of dendrites in the control mouse versus the stressed mouse in the layer 5 of the pyramidal neurons. And you can see that there's much less of the connections or synapses or synaptic things that spines, I think he calls them. And this is, I believe, in the prefrontal cortex. Yeah, the medial prefrontal cortex. And on the right, in the stressed animal, there seems to be a loss of synaptic spine areas. And the other thing that can affect this is the state of BDNF, which is brain-derived neurotrophic factor, which can exist as polymorphisms with where a valine, it can be substituted or changed to a methionine. And what happens here, if you have the native form, as shown here, which is of the two alleles that both have the valine in position, you get a kind of a lot of dendritic, synaptic-looking spines and normal-looking connections. If you have one allele that's changed, you lose some of that. If you have both alleles that show that polymorphic change, you're losing a lot of your dendritic spine areas, and it results in as well. And when that's changed, it causes it decreases the transport of the BDNF transcripts and the activity-dependent release of the BDNF as well. So he does another example of a stress model on the next slide, which he what he does here is stress the mice with what he calls chronic unpredictable stress for 21 days. And so again, looking at these spine synapses is what he's calling them. In the upper layer, in the control, you can see there's a fair number. And then in the middle layer, after 21 days of stress, it's reduced. Then if you give them ketamine for 1 day, 24 hours, it restores it back to normal looking. And you can see that in a number of other parameters. Below that, he's looking at EPSPs, excitatory postsynaptic potentials, induced with either 5-HT or hypocretin. And you can kind of see that there's less EPSPs induced in the mice that underwent stress, but when you add ketamine, it comes back. And what's listed here below that, C and D, is just a quantitation of what you see in the middle. And so the synaptic or synaptogenesis seems to be restored by ketamine in 1 day. And then if you look at a behavioral consequence of that, and that's in E and F at the bottom, what he's showing is two things. The one on the left, bottom left, is E, is a model for anhedonia, basically. What he's looking at is the preference that the mice might have in terms of percentage preference for a sucrose solution. And the stressed mice that are depressed show anhedonia because they don't go to the sucrose solution as much as the control mouse, but when you add ketamine, it comes back to like the control. And the one on the right at bottom, F, would be a model of anxiety. And what he's looking at here is the latency to feed in an open field. And so if it's an anxious mouse, they're not going to be quick to go into an open field. They're going to be very hesitant. So it's going to take longer till it feeds itself. And that's shown in the middle. The stressed mouse is taking a longer time to feed than the control, but if you add ketamine, it comes back to something that looks like control. So what part what might be going on here is shown in his model slide from 2012. And what he's saying is that there's it in part it has to do with the AMPA. What he's showing here is the AMPA receptors. There are three kinds of glutamate receptors, which I think you know already. The NMDA, N-methyl D-aspartic acid receptor. There's an AMPA receptor and a kainate receptor. But what he's showing here is in a regular state of mood, the AMPA receptors are on the surface of the postsynaptic neuron. And under conditions of stress, they disappear from the surface. So you lose that synaptic pathway because the subunits of the AMPA receptors get internalized. And that's controlled by GSK3, which is glycogen synthase kinase 3. When GSK3 is dephosphorylated by PP1, protein phosphatase 1, it becomes active, and it in turn can phosphorylate the AMPA receptor subunit and cause it to internalize. So that's one part of it. And so what they find is when you add a rapid antidepressant like ketamine, as shown on the right side, you restore the AMPA receptors back to the surface of the postsynaptic neuron, and how that probably works is that the ketamine, as we'll try to explain a little bit later, the ketamine is an antagonist of the NMDA receptor, and by doing so, it can lead to increased synthesis and release of BDNF, the brain-derived neurotrophic factor, which in turn, as shown here on the right, the released BDNF acts back on its receptor, um, which is track B, and that's it's a tyrosine kinase receptor. It stands for tropomyosin receptor B kinase, and that's the actual receptor for BDNF, and that triggers a signaling pathway, which causes an activation or increase of AKT, which is another name for protein kinase B, which in turn phosphorylates mTOR, or the mammalian target of rapamycin. And the mTOR influences, it's not shown here, but it influences the EEF2, which is eukaryotic elongation factor 2, which increases protein synthesis of BDNF and some other proteins, and the BDNF is then released. And the AKT also can inhibit GSK3, preventing the internalization of the AMPA receptor. Okay, so that's these are the figures from that paper we wanted to cover as a journal club. So I'm going to digress a little bit, but I'll try to keep this relatively short, I think. But there was something else I mentioned to you at the beginning of the ILO, which had to do with the work I was doing many years ago on PDEs. I still work on them. It's phosphodiesterases, or cyclic nucleotide phosphodiestrases. And I just want to mention this. So I actually first purified what we know now to be PDE4 40 years ago. In 1979 is when I first published on it and then characterized it. I actually had a position at University of Texas Medical School for 4 years, from 1975 to 79, which is where I was doing the work. Just to let you know, I didn't. I was purifying these from dog kidney, but I didn't kill any of these dogs. The way this came about is I found that this enzyme was highly expressed in brain and in kidney and in lymphocytes. And there was the late Tom Burke was another faculty member who had a lab down the hall who was working on opiate dependency using dog models. And he would go through a dog a day and take the brains, and he was throwing the rest of the dog out. So I just kept stockpiling the kidneys until I had enough to try to do a purification. I've always had dogs and cats all my life, so I wouldn't want to harm them. But there was nothing I could do to stop him from doing that research, so tissues were available anyway. So I purified this, and then I brought the purified enzyme with me here. I came here in May of 1979 and then continued to work on it and characterize it in catalytically amino acid sequence, and then I looked at the sensitivity of a whole range of pharmacological agents, and that enabled the fact that I purified this and did that enabled a fellow in Germany, a Helmut Wachtel from Schering Company, to develop a selective inhibitor for this enzyme called Rolipram, which I had mentioned to you before. And as shown on this slide, he entered this into clinical trials as an antidepressant, and it went head-to-head with a couple of tricyclic antidepressants, imipramine and desipramine, and seemed to be equally effective in clinical trials. But the problem was essentially that the PDE4 selective inhibitor caused emesis and nausea, so it couldn't get approved, and it's been difficult to separate the efficacy from that side effect even now. So I don't know if the PDE4 inhibitors will make it or not, but he wasn't too clear on how this worked. He thought it had something to do with increased norepinephrine turnover, but later, toward the end of the 1980s, when there was more interest in BDNF and its discovery, the same fellow whose paper we just covered, Ron Duman, was pointing out that the hippocampus in depressed people had atrophied, and he felt that that was probably due to loss or decrease in BDNF. So I came up with a model for how the roloprane was working. I pulled this next slide here out of a talk I gave about 20 years ago, where I believe what's happening is that the PDE4 the roloprane by inhibiting the PDE4 increases the cyclic AMP level, which acts through CREB, a transcription factor that induces the BDNF in the hippocampus. That's the cyclic AMP receptor element binding protein that drives the transcription of BDNF. So that's probably how it's working. But so hopefully maybe we'll get one of these eventually to work as a without too bad side effects, and might prove to be another effective antidepressant. But in the meantime, the reason I bring this up is I keep working on PDEs, and we didn't know how many there were, which ones were what, and until we started doing cloning. We didn't have the technology or the techniques to clone these at that time. It wasn't until 1994 that I first started cloning the PDEs, and then we realized after that there were 21 different genes encoding PDEs, and could separate them into 11 gene families. And this PDE4 we called it the high affinity cyclic AMP specific PDE, but then in the early 2000s, I found, and other people found, there was another cyclic AMP specific PDE with a much higher affinity, about 100-fold higher, a km of about 40 nanomolar, and we designated that PDE8 family. And so I started doing quite a bit of work on PDE8. In particular, what happened is in 2004, when Stefan Brock, who's here in the immunology department, came here, he had given a talk right after he came about his work, which involved looking at transendothelial migration, or how activated lymphocytes cross the blood-brain barrier in these EAE models of multiple sclerosis in mice. EAE stands for experimental autoimmune encephalomyelitis. These animals show symptoms of MS, and he was trying to figure out how the lymphocytes get across the brain and trigger this inflammatory response in the brain. So I went up to him afterward. I said, there's a lot of evidence that the cyclic AMP signaling pathway can control cell movement, and it would be interesting to look at the PDEs and see if we can find whether one of them or another is controlling that movement of the activated lymphocytes. So I did a study and found, in fact, concluded that PDE8 was responsible for controlling lymphocyte chemotaxis. And we published this in 2006. I gave a talk at an international Gordon conference. It was held at University of New England in Maine, and it attracted a lot of interest. And a fellow named Frank Minetti came up to me right afterward. He was a senior member of the neuroscience department at Pfizer, and he said he wanted to try to collaborate with us on it, and that they ended up making a very potent selective inhibitor of PDE8, which they gave us. Stefan was then able to go further with this with a couple of his graduate students here, showing, with Amanda Vang, showed that by inhibiting PD8, it also inhibited the adhesion of lymphocytes to the endothelial cells, which is the first step in transmigration. And then he had another student, Shaitoli Basali, who did work with the EAE mice and showed when you treated them with this PD8 inhibitor, it reversed the symptoms associated with MS. So we concluded that PD8 is maybe a tremendous target for treating autoimmune and inflammatory diseases. I had also shown back in 2006 that PD8 gets induced when lymphocytes are activated. So we're still concentrating now on PD8 in a lot of our work. And then the reason I bring it up is because of this next paper that just came out this year. It's a very interesting paper. If you're interested in learning more about PD8, we published a review on it that came out on April 16th, 2021, and some information on it and a link to it is here. It's known to be as shown on this slide, it's known to be a antagonist of the NMDA glutamate receptor. The NMDA receptor is a complex receptor that's controlled in a few ways. It's an ionotropic. It's ionotropic, and it has passes non-selectively. Can pass bivalent cations, or just cations, mono or bivalent. So the glutamate, there's a site to which glutamate binds, is shown in the upper left, and it also requires the binding of an amino acid in another site, and that could be either glycine or D-serine. And it's also basically shut down because magnesium is binding directly in the channel, or it could be zinc as well. Either extracellular magnesium or zinc binds tightly in that channel. So even if these two things bind to it, it's not going to pass any other cations. It also requires it's voltage sensitive, and it requires some depolarization to knock the magnesium out of the cleft so that the other ions can flow through. And what happens is the potassium goes out and sodium and calcium come in. And what happens is ketamine will bind to the same site that magnesium was binding to. So it will the receptor has to be activated first by depolarization to remove the magnesium before the ketamine can bind. So the next slide is just the structure of ketamine. At physiological pH, it's positive. The amino group in ketamine is positively charged, and it binds where the magnesium was binding. It's sometimes referred. It's sometimes called a non-competitive antagonist because it's definitely not binding where the glutamate is. You can't reverse it with increasing glutamate, but more often it's actually referred to as an uncompetitive antagonist. What that means is it's not going to antagonize that NMDA receptor until it gets activated by depolarization and the magnesium is removed. And that's why it's considered sort of called uncompetitive. And it has a KD for binding of about 0.5 micromolar. So then the next couple of slides show how we think this is working. You already saw some of it from that last slide of Dumont's paper, but been other reviews. And this next slide is from this Nature Review article by Murrow. And what he's showing here, unfortunately, he doesn't have everything here, but if you look at the postsynaptic pyramidal neuron here at the bottom, there are three types of glutamate receptors here, the AMPA receptor, the NMDA receptor, and the kainate receptor. Unlike the NMDA receptor, the kainate receptor and AMPA receptors don't require a second allosteric site for glycine, and they don't require voltage depolarization, or they're not blocked by magnesium. But in any event, what I want to get at is that there are different NMDA receptors. These are the ones that are in the postsynaptic cleft. There are also some that are extrasynaptic. If you look at the bottom right, that's an example of one here with that number 7. And this is what he's missing. There are many of them in the presynaptic receptors as well, and in the GABAergic interneurons. And what's believed to be happening is that the release of glutamate from this presynaptic receptor is somewhat tonically controlled by GABA. Okay, GABA obviously would inhibit its release. And it's not shown here, but the NMDA receptor that might be near there in the presynaptic neuron, NMDA receptors are excitatory receptors that would drive the GABA, which would be inhibiting the release of the glutamate. So if you come along and you inhibit that NMDA receptor in the presynaptic neuron, you would basically not release so much GABA. Therefore, you'd release more of the glutamate from in the cleft area, and you would then activate these postsynaptic glutamate receptors like the AMP receptor and the postsynaptic NMDA receptor. Now, the important thing to know here is that the NMDA receptors in the postsynaptic cleft differ from the ones extrasynaptically and the ones that would be in GABA interneuron presynaptic regions. They have a different subunit composition and different sensitivity to ketamine. And what's used for antidepressant is what's called a subanesthetic dose of ketamine, 0.5 milligrams per kilogram. If you use a 1 to 3 milligrams per kilogram and you have an anesthetic dose, it does not work as an antidepressant. The reason being that the synaptic NMDA receptor is less sensitive to ketamine, so at the low subthreshold doses, the extrasynaptic NMDA receptor and the ones in the GABA interneuron are inhibited by ketamine, but the one in the synaptic cleft is not. And so the glutamine that comes out can still activate it, and it will activate the AMPA receptors. That will drive more calcium to come in. The calcium will then cause the release of BDNF, and that will act back on this TrkB receptor that I mentioned, the tropomyosin receptor B kinase. That in turn phosphorylates a signaling cascade using ERK and the AKT, or protein kinase B, which acts on another protein which he left out here, unfortunately. It would be near the number 9, but it would be the mTOR, which is a mammalian target of rapamycin. There's another kinase, and the mTOR in turn acts on EEF2, which I mentioned is the eukaryotic elongation factor 2, which increases protein synthesis. So you make more BDNF and other structural proteins involved in synaptogenesis. So that's kind of what's going on. And this same review shows in the next slide the spots where ketamine might work. So in the very upper part in the right, it sort of shows it like the NMDA, the ketamine is inhibiting the GABA. It's not directly inhibiting the GABA receptor here. It's just inhibiting that NMDA receptor that's located in the presynaptic neuron. And by inhibiting that, the GABA is less active. You get less GABA, because it's not being activated by that receptor, so you're getting less GABA out. And then, as he shows in number 2, as a result, ketamine is actually, by doing this, it actually drives more of the glutamine to come out and activate these postsynaptic NMDA receptors. At the same time, the ketamine will be inhibiting this NMDA receptor extrasynaptic one, which normally is blocking the elongation factor, but by inhibiting that, it takes away that inhibition. And again, the release of more BDNF acts through this TrkB receptor cascade to release more BDNF, so it's a kind of a positive loop cycle, and you get more synaptogenesis. The next slide is similar. It's just from one of Ron Dumond's recent review articles. And here you can see he's got the NMDA receptors in the GABAergic interneurons, and those are very sensitive to inhibition by ketamine. And what happens in cases of with chronic stress and depression, as is shown here, which is the one on the left, you get decreased GABAergic function, and you're going to get way more activation of these glutamate channels and much more calcium coming in, and it's a condition we refer to as excitotoxicity, which seems to be associated with a number of neurodegenerative diseases, but it leads to that increased calcium leads to atrophy or apoptosis of these neurons. So you get neuronal loss and spine loss, which we had seen in his earlier slides. When you add ketamine, you reverse the whole process. It's by the mechanisms we just showed you, by increasing more BDNF. So this next slide, it's just the same thing, showing the places where some of these rapidly acting antidepressants can work through these systems. What's of note here is, if you look on the left, there's the one mentioned here, brexanolone, and we talked about that briefly earlier in the depression lecture earlier, because that compound was approved as well as ketamine was approved also in March of this year, specifically for postpartum depression, and that agent also works fairly rapidly. It has its full effect within 48 hours. And the brexanolone is a progesterone analog which acts on the neurosteroid site directly on the GABA-A allosteric site on the GABA-A receptor. And so it's also having an effect on this system. All right, just very quickly, this next slide just is simply showing that as of 2016, when esketamine was in clinical trials, which is now approved, there are quite a few other agents that have been and are in clinical trials. Some of them working on some specific subunits of the NMDA receptor, like the NR2B subunit. So it's likely that more compounds will come out, some of which may have better pharmacokinetic properties or duration of action. The thing with ketamine or esketamine is it works immediately, but it only seems to last for about a week, and you need repeated treatments of this to treat the depression. Finally, I just want to show you another experiment I did 40 years ago. It's here. When I first came here, there was the fellow who was the chair of pharmacology, Bob Vole. He was the chair of the department from when the first medical class started in 1968, and he spent most of his career studying the nicotinic acetylcholine receptor, but he was an electrophysiologist, and he had some evidence that ketamine seemed to interact with the nicotinic receptor channel, and in an active state of the channel. So when I came, he asked me if I could do any biochemical analysis that might support his contention. So what I did is I isolated these microsacs from the electric organ of a torpedo fish or an electric ray fish. These are they're highly enriched in nicotinic acetylcholine receptors. These fish can deliver a charge of 220 volts to its enemies. So I isolated this and I set up tritiated phencyclidine binding, or PCP, because that's an analog of ketamine that I knew bound to the nicotinic receptor. And then what I found is um, the ketamine could displace the binding of the PCP with a Ki of about 42 micromolar. That's the dotted line here. But if I added carbachol, which is an analog of acetylcholine that's non-hydrolyzable by acetylcholinesterase, so it sticks around longer. I added that to open these nicotinic channels and to the open state, and then I saw a tremendous shift to the left, and the Ki was about sevenfold. It was about 6.6 micromolar, about sevenfold higher affinity of the ketamine in the active state of the nicotinic receptor, which supported his contention. So we had evidence that ketamine could also act on something like the nicotinic receptor besides the NMDA receptor. And I hadn't followed this field since. This was like, as I say, almost 40 years ago. But just this week, when I was trying to look for a structure of ketamine to post on this for you guys, I went to Wikipedia. That's where I pasted the structure from, and this is what I found in that ketamine version of the Wikipedia. They show apparently the ketamine binds to a whole slew of other types of receptors, not just the nicotinic receptor like I showed. In the next slide, he actually shows the affinities. The one that's sort of about midway down where it says alpha 7, that's the nicotinic. That's a receptor for nicotinic acetylcholine. And wherever he was getting this from, they get an IC50 of about 20 micromolar, which is close to, I was getting 40 or 42 in the absence of carbachol, so they're fairly close. So it doesn't matter that much, but because if you look at this, almost all of these that it binds to, it's binding with a weaker affinity than for the NMDA receptor itself. So we probably won't see a lot of side effects from this unless the ketamine gets too high. But it's something to consider. Anyway, that's it. Okay, I hope this gives you an idea of something about the way that this ketamine works. And you'll see more of these compounds coming out. Okay, thanks.